A very good morning to one and all. This is S.Vaidehi, Assistant Professor working in DG Vaishnav College, Chennai. And now I am going to present on the topic Bioremediation Potential of Bacterial Strains for Decolorization of Malachite Green and Crystal Violet. Water is essential for the survival and continuity of life. We all know the water is now got polluted due to industrial growth and insufficient wastewater treatment. The major industrial sectors which leads to water pollution are textiles, leather, paper, printing, food processing industries, from which the large volume of waste is released into the effluent water bodies. And the major industry which is involved in major contribution to water pollution is textile dyeing industries. The dye which are secreted from the textile industry directly enters into the water, and the dyes which are released which are used in the textile dyeing industry are classified approximately into 20 to 30 groups, which includes azo dye, anthraquinone, phthalocyanine and triaryl methane dyes. Azo dyes accounts for nearly 60 to 70% of global dye production due to their stability and low cost. While using dye for textile industry or to dye the clothes, 10 to 15% they doesn't bind to the fibers and they are released into the wastewater. Presence of such dyes in water, they reduce the light penetration into the water, and they also interfere with the photosynthesis, and thereby they negatively affect the aquatic organism which is present in the water bodies. Especially dyes such as crystal violet and malachite green, they are of major environmental concern due to their widespread use in textile industries, aquaculture and veterinary applications. In general, the properties of dyes which are used in textile industry are chemically stable, polybiodegradable, and they exhibit significant toxicity. The toxic effects of the major two dyes which are listed earlier, crystal violet and malachite green, is represented here. Crystal violet is highly cytotoxic, genotoxic, mutagenic, and it is also a potent carcinogen. Malachite green is teratogenic. Again, this is also mutagenic and carcinogenic. Apart from that, it is also going to cause pulmonary toxic effects. In general, it is also going to cause mutation by chromosomal damage. There are different treatment methods which are available to treat the water which are polluted with dye or with any industrial waste. The first thing is the physicochemical method, which includes adsorption, chemical precipitation, flocculation, oxidation using chlorine, hydrogen peroxide or ozone, and electrochemical treatment. Though these methods can remove color effectively, but they are very expensive, and apart from that they are going to generate a large amount of sludge which cannot be broken down. Next, coming to the conventional biological treatment methods, whereas the activated sludge process, they reduce the BOD and COD content in the water by removing the dyes, but the dyes are very resistant to microbial breakdown, which is biosorption. So the one major method which can be employed to treat water pollution is the biodegradation or the biotransformation, where the microorganism is going to release some enzymes that will break down the dyes. Materials and methods. For this study, pure culture of E. coli, Pseudomonas and Bacillus were obtained from the microbiology department, and they were grown in nutrient broth. To get another microbial consortia, all these mixtures were mixed together and they were inoculated. The dye decolorization assay was carried out to find out the bioremediation properties of the microorganisms. Nutrient broth were pipetted out into different test tubes, and to all the test tubes, different concentrations of the dye malachite green and crystal violet were pipetted out, and the tubes were inoculated with the bacterial cultures like E. coli, Pseudomonas, Bacillus, and mixed bacterial consortium. All the experiments were carried out in triplicates. The broth was mixed well and they were incubated in incubator at 37 degrees Celsius. At regular intervals, the absorbance of the dye, which was reduced by the microorganism, were measured. For crystal violet, the UV absorption spectra was read at 570 nm. For malachite green, it is 620 nm. Using the formula, the percentage of decolorization was calculated. This graph represents the microbial degradation of the dye crystal violet. This study was also carried out for a period of 7 days. Among all the individual isolates, E. coli exhibited highest decolorization efficiency, followed by Bacillus and Pseudomonas. Microbial consortium removed around 80% of the dye over a period of 7 days. This study also supports that all the microorganisms, they shows a time dependent increase in dye degradation. Conclusion: the findings of the present study demonstrate that the bacterial strains E. coli, Pseudomonas, Bacillus and their mixed consortium possess significant potential for the biodegradation and decolorization of malachite green and crystal violet dye. This may be due to the presence of lacase or the release of lacase and its action on the dyes. The progressive reduction in dye concentration over the incubation period confirms that the removal was predominantly mediated by biological activity. Notably, the enhanced performance of the microbial consortium underscores the importance of synergistic interaction in the degradation of structurally complex dyes. Collectively, these results support the applicability of bacterial system as effective and sustainable tool for the treatment of dye contaminated and